Hello, everyone. Good afternoon. Before I introduce our speaker, I want to see if anyone has any announcements. Cleveland, you usually have an announcement. Okay. Any, Professor Mullen, do you have an announcement? The announcement is, where is everyone?
My announcement is that we have a chosen T-shirt person for next year. It was a very tight vote, but Gary O'Neill's octopus and anchor was the winning design. I'll send an email out to it, but he's not here right now to celebrate his winning design.
Congratulations, Gary. Oh, I thought they were coming for the seminar. Okay, so I am very happy to introduce our seminar speaker today. So Dr. Dave Ernst is joining us from Bigelow Lab in East Boothbay, where he is a research scientist. He's from Illinois and got his Bachelor's of Science in Animal Sciences from the University of Illinois, and then went to University of North Carolina, Chapel Hill for his PhD, where he studied navigation and sensory biology of migrating marine animals. Super cool. Specifically spiny lobsters and sea turtles. And then after grad school, he was a postdoctoral researcher at the University of Arkansas, where he used molecular techniques to understand the sensory ecology of butterflies. And then from there, Dave moved to Bigelow Lab to study shellfish larval ecology using environmental RNA. And so that's the work that we're going to hear about today. So thank you, Dave, for joining us, and I look forward to your presentation. Thanks.
Can you all hear me? There we go. Good. All right, I'm going to go ahead and set the timer here. Well, thank you so much for that introduction, Leanne, and thank you all for coming today. I feel privileged to be able to come here and speak to you all. So thank you for the opportunity and for the invitation. And so as Leanne mentioned, today I'm going to tell you about some of the work that I've been doing here at Bigelow as both a postdoc and more recently as a research scientist. And so I'll circle back to a little bit more about me to start off with. And I know Leanne covered some of this, but I'm originally from Central Illinois. I'm from a pretty small town, for someone that studies marine biology and ecology, pretty far away from the coast. But I did have kind of these seas of corn and soybeans surrounding me growing up, and really just always kind of had a fascination with the ocean and the creatures that inhabit the ocean. From an early age, I kind of knew I wanted to do something with marine science. And so, as Leanne mentioned, I went to University of Illinois for my undergraduate education. And this is where I really got my first taste of research. I worked in Allison Bell's laboratory up here on the upper right-hand corner that you see. And most of the research in her lab was focused on this really small fish that you see right here in the center called the three-spined stickleback. So this is a fish about 2 to 3 inches long. Really great kind of evolutionary biology and animal behavior model. And so I spent the better part of 2 years studying three-spined stickleback behavior and had the opportunity to go out into Northern California to study them out in the wild and spend some time out in the coastal redwood forests camping for a couple summers doing that field work. And so this really is another thing that got me interested in continuing on with research and field work specifically. And then next I moved on to University of North Carolina at Chapel Hill, working in Ken Lohman's lab up here on the upper right-hand corner. Here I studied navigation in marine animals, but specifically I was studying this pretty unique sense called magnetoreception. This is essentially just the ability for some organisms to detect Earth's magnetic field and then use that information to establish a migratory heading. So this is really probably best well known with birds, but sea turtles are also very well capable of this. And I spent most of my time working with the Caribbean spiny lobster on the lower left-hand corner that you see here. And so during the latter half, really the latter quarter of my PhD, I started asking questions that required a molecular biology skill set. And so I started getting into things like genomics and some other molecular biology techniques, and really realized kind of the power behind those, which led me to my first postdoc. And so this may seem kind of strange in that I started off in Illinois really hoping to work with marine systems and study marine biology. I go out and I finally make it to the coast, and then I come back to the center of the country. And so I'm at the University of Arkansas for my first postdoc here, and I'm studying a few terrestrial species, so some African and Central American butterflies. But really the reason that I did this was to build upon my molecular skill set. So I just wanted to say, for anybody that is interested in continuing on to graduate school and pursuing a research career, postdocs are a great opportunity to expand your skills or expand your research horizons beyond what you've done in graduate school. And so this is a pretty hard shift into a brand new system and asking very different questions that are focused on much more of the neuroscience side of things. All right, and then from there, I finally got back to the East Coast, where I currently am at Bigelow Laboratory for my second postdoc, and I joined Bigelow Laboratory as part of the main eDNA project. And so this is a big NSF EPSCoR grant. There's a 5-year grant that actually just wrapped up this past December, and Leanne was a part of this. And this is really aimed at trying to establish Maine as kind of one of the epicenters for environmental DNA research, as well as to develop environmental DNA tools to better understand Maine's coastal environments. And so I joined as part of Nicole Price's lab, working on what we called the larval black box project. And this really aimed to better understand kind of these shellfish larval distributions throughout the Gulf of Maine. Oh, I should mention, this past September is when I moved into my current position as a research scientist, continuing on with some of this research. All right, and so before I jump into that research, I just wanted to make a note that, as you may have noticed, that career paths are very rarely a straight line, right? So I've jumped around quite a bit, working in a number of different systems with a bunch of different, trying to answer a bunch of different questions. Just wanted to say, for those of you that are interested in pursuing a career in research or just a career in general, you'll often find yourself moving in unexpected directions, but you almost always get to where you're trying to get to. All right, and so to jump into the work that I'm currently doing and the work that I've done as part of this main eDNA project, I am first going to start by talking a little bit about mussel aquaculture here in Maine and some of the current challenges that it's facing. And this will kind of frame some of this environmental RNA work that I'm going to tell you about today. So mussel aquaculture here in Maine, and really across the world, so Europe, New Zealand, China, they really are dependent on, or the industry is really dependent on capturing wild larvae from the water column. And so you see here right in the center picture, this is just a piece of frayed rope, they often call fuzzy rope. These will be placed in the water column, and the mussel larvae will settle upon those ropes, and then they'll essentially suspend them on these mussel rafts, which I'll talk about in just a second, and allow them to grow out to market size. But it's really key that they're actually able to capture this, what they call mussel seed, in the first place. And so to take a step back and just walk you through quickly the life cycle and kind of these stages that we're really interested in. So this is the green shell mussel, but this is essentially the exact same life cycle of the blue mussel. Mussels are broadcast spawners, meaning that they release both eggs and sperm into the water column. Those eggs are then fertilized within the water column, and those fertilized eggs rapidly develop into the first free-swimming larval stage, the trochophore. This is a really short-lived stage. It's only about 24 hours or so, after which it develops into this, what they often call a D-stage veliger. It kind of looks like the shape of the letter D. That's where it gets its name. And then finally into the pediveliger stage here, where it starts to develop that characteristic foot of many bivalves. And this is usually a couple weeks that they'll spend in the water column. After this, once they reach this late pediveliger stage, they'll then settle and metamorphose, or they'll find a settlement substrate, I should say, like the seagrass here, and then they'll metamorphose into a post larva, or what they call spat. And then they'll grow out into juvenile and adult mussels. And so this late pediveliger stage and this spat stage is really what mussel farmers are really interested in. And this is just a picture of some of these rafts that I was talking about earlier. What we use in Maine is typically called suspended culture. And this means that you have these rafts that have these cross beams, these wooden cross beams that you see all over here. And then you can see, it might be a little bit difficult to see, but there are ropes hanging down from each one of those cross beams. On those ropes, you've got tons and tons of mussels. So this is a diver here that is holding onto a couple of those ropes on just below one of these rafts. You can see they're absolutely just covered with these mussels. These mussels are attached to the ropes with their byssal threads. So this is just a picture here of them pulling up one of the ropes here that's covered in mussels. Now, one big challenge that the aquaculture industry has been facing is that mussel seed has become really unpredictable. Many farmers used to be able to throw out their seed collectors, which typically consist of those frayed fuzzy ropes that I mentioned, maybe the second week of June or the first week of July. And consistently, they're always able to capture that seed. However, in the past 10, 15 years or so, that story has changed. Many mussel farmers have found that the seed supply is becoming more and more unpredictable, and they're starting to have recurring recruitment failures, meaning that they're unable to capture the seed that they need to produce their product. And this could be due to a number of different reasons. I've outlined a few of them here on the bottom left, but what's clear is that failing to capture the seed in a given year can have very severe impacts for a farm. They won't be able to produce product, and then that will lead to severe financial impacts as well. So this is a really big problem that needs solving here in Maine and elsewhere. So how can we minimize these recruitment failures? Well, one way is through hatchery cultivated seed. So we can just grow the seed, right? And then supply that to all the farmers, and all should be good. However, well, first I should mention, we do have a number of people doing this here in Maine, including the Down East Institute. They've got a fantastic hatchery facility, and they're making a lot of great progress. But still, with all the work that they've done, they're not able to supply all the seed that would be needed to support the current farmers here in Maine, not to mention the future farmers as this industry grows. And one of the big bottlenecks here is it requires a very significant investment in infrastructure as well as workforce. It's really labor intensive to do this kind of work. And so as of right now, this isn't really a viable solution here in Maine. So as an alternative, we can actively monitor mussel seed within the water column. And surprisingly, this hasn't really been done by many farmers, hardly at all, except for the past couple years when these issues started to arise. And I guess perhaps that makes sense because there hasn't been a need for it, right? They've always been able to capture that seed. And so what I mean by active monitoring is you could simply go out and take a plankton tow, which I'm assuming probably a lot of you have done in your courses. And then take a look at that plankton tow under a microscope, and then you can look here on the bottom right-hand corner and just enumerate the bivalve larvae that you see. However, there is one major issue with this, and that is that bivalve larvae of different species are extremely morphologically similar. And so unless you have specialized equipment or really specialized training, if you're trying to do this on the fly for a tow that might have mixed species of bivalve larvae, your counts are going to be way off. And so I'll just ask, can anybody here pick out the mussel larva? Any takers? No? What was? H. Oh, close. It's E. Very close. Yeah, as you can see, just looking at this, these are all different species, and this one is the mussel larva here. And so one way that we can get around this problem is through using molecular tools like environmental DNA and environmental RNA-based tools. Doing this will allows us to use things like digital PCR, quantitative PCR, and what we call a LAMP-based assay. All of these I'll talk about a little bit more here in a few minutes. But it allows us to detect specific molecular signatures, like a specific sequence for an individual species, to definitively determine whether or not that actually is a mussel larva. All right, so taking a step back, what is environmental DNA? Most of you have probably heard of this. This has become a lot more popular in the news and in just research in general, and it sounds like you've heard some about it in some of your classes. This is just DNA that's present within the environment, exactly as it sounds, right? Or within an environmental sample that you take. And so this can be in the form of free DNA just floating around in the water. It can also be cells or skin or scales, waste products from organisms that are living in that water or passing through, respiring. Or it could be entire organisms in the case of bacteria or small planktonic larvae, for instance. And what's really cool about this is, as I just mentioned, we can design assays using qPCR, for instance, that are really targeted for specific species. So for instance, if we wanted to detect this specific fish, we could design an assay for that, and then we could detect that fish and then get an idea of the abundance of that fish. An alternative approach that we can take is using next generation sequencing and using a technique called metabarcoding. And basically what this does is it targets a gene that is conserved across a ton of different species, in this case, fish species. And in the middle of that gene, there's a region that's really, really variable and acts almost like a barcode, hence the term metabarcoding. So there's a barcode for each different species, for instance. And what this allows you to do is look at kind of the whole host of taxa that are present in that sample and get an idea of the relative abundance. Now, how about eRNA? What am I talking about when I say that? Well, it's very similar to eDNA, except for the substrate is just changed from DNA to RNA. And when I'm talking about RNA, if we revisit the central dogma here, I'm talking about expressed messenger RNA transcripts. And so we've got a DNA strand up here, and mRNA is transcribed from that DNA strand, and then that mRNA is translated into a protein. And so to simplify this a little bit more, you can think of DNA kind of as the cookbook that contains all the instructions or recipes for all the proteins that an organism needs. You can think of mRNA as copies that are made of that recipe. So for instance, maybe the chef is making a bunch of copies of the recipe for a particular protein and handing that off to the sous chefs. And then those recipes can then be taken to make the final product, the proteins or the dinner in this analogy. And so really what we're going to be focusing on here is this, this middleman or this messenger RNA. So you're probably wondering why on earth would we want to use environmental RNA over environmental DNA? And there are three key advantages of environmental RNA over DNA. Environmental RNA tools are capable of detecting metabolically active organisms, and by that I mean living organisms. So RNA is only produced by living organisms, whereas DNA can be sloughed off from decaying organisms on the seafloor, for instance. It offers a greater spatiotemporal resolution due to its lower stability relative to DNA. And this is because RNA is a single-stranded molecule. So this gives you a better sense of kind of what's in your sample here and now. Whereas eDNA, or just DNA in general, lasts a lot longer, has the potential to travel much further distances than RNA. And then finally, and perhaps most importantly for the work I'll talk to you about today, is it's capable, these tools based on environmental RNA are capable of subspecies level resolution. And what I mean by that is we can get at different life history stages or developmental stages. We can look at physiological state. Are these organisms really stressed out? Are they about to die? Things like that, which is not possible with our current environmental DNA tools. Or this is made possible with environmental RNA because all kinds of genes are turned on and off over the course of development. And so you can imagine a gene that might be really important for larval development, might be really highly expressed in larvae, but show no expression in adults. And what this does is it gives rise to these larva-specific transcripts. And so, effectively, if we have a way to detect those transcripts that are only expressed in larvae, then we have a way to specifically detect the larval stages of blue mussels. All right, so had four main goals for the work that I'll talk to you about now. One was to develop environmental RNA-based tools that are designed to detect and determine the abundance of blue mussel larvae in water samples, to test those collection kits as well as the assays on an operational mussel farm here in Maine, in the Gulf of Maine. Then to get these kits in the hands of some of the mussel farmers so that they can test them out, tell us what needs optimization, what works, what doesn't, are they likely to even use them? And then finally, and ultimately, I guess, we want to use these to not only enhance our understanding of the mussel larval dynamics within the Gulf of Maine, but we want to be able to inform seed collection for mussel farmers and effectively enhance domestic production of mussels. So I didn't mention this before, but vast majority of our mussels are coming down from Prince Edward Island from Canada. And so this will help to kind of boost the production here in the US. All right, so great. This sounds good, but how on earth do we do it? The first thing that we need to do is collect samples from all the different life stages so that we can get a sense of what gene expression looks like across development. And we're very fortunate to have the Down East Institute here in Beals, Maine. They have a really fantastic hatchery facility. For most of the year, they are they're spawning blue mussels, I think every 2 weeks now, which is pretty incredible. And so this gave us a great opportunity to collect samples from each of these different developmental stages. And so I just want to give a shout out to a number of people that helped immensely with this. So Brian Beal and Kyle Pepperman, but especially Bree Salter and Elsie King, who actually did most of the collection on these for us. And also I want to give a shout out to Leanne and Aubrey Mitchell, who is a previous MMA student and an REU intern for us for 2 summers, I guess, that worked on this various aspects of this project as well. So don't want to forget to mention them. All right, and so we collected both gametes, so sperm and eggs, the trochophore stage. This is that D. veliger that I talked about a little bit earlier. The pediveliger stage, you can see the foot right here. Post-larval stage, after they've settled and metamorphosed, and then finally adults. We collected both male and female adults in case there were differences there as well. All right, so now that we've got all these samples, what do we do with them? Now we need to sequence them using RNA sequencing and then characterize the transcriptomes. And what I mean by transcriptome, this is just the collection of all those messenger RNA transcripts that are present within a particular developmental stage. And so once we sequence all those mRNA transcripts, we can then align the reads that we get back to the blue mussel genome, and this allows us to quantify gene expression and transcript abundance. All right, and what you're looking at here is just kind of a really high-level overview of the gene expression profiles of these various stages. And so each one of these points that you see here indicates the gene expression profile of one individual sample. The closer that those are to one another, the closer those points are to one another, the more similar they are. The further apart they are, the more different they are. So this is forgot to mention, a principal components analysis plot here. And what's really cool about this is that we see that each one of these developmental stages clusters independently of the others, meaning that each one of those stages has a distinct gene expression profile. And so we were really excited when we saw this because this means that there's likely a lot here that we have to work with in developing these environmental RNA tools that are reliant on these gene expression profiles. So there's some major differences between the stages, in other words. And looking at this a little bit deeper, this is a plot called an upset plot, probably because it's somewhat upsetting to look at and try to make out what's going on. But what I want you to pay attention to are these red dots here. You can think of this as just kind of a Venn diagram for a lot of different samples, so 8 different samples here. Each one of these red dots and the bar above it indicates how many genes showed gene expression or had genes that were only expressed in that given stage. So for instance, right here, this is eggs. There are over 5,000 genes that are only showing expression in mussel eggs. So we essentially have a lot to work with here. We've got a lot of genes that show stage-specific expression. We know that these expression profiles differ very much. So now that we've got these candidate gene targets to work with, we then started to design quantitative PCR assays, which consist of a couple primer sequences and a probe sequence that are designed to detect these different targets. And we specifically made them so that they only amplify the messenger RNA transcripts and not genomic DNA. So this allows us to specifically get at RNA and avoid any DNA contamination. So now that we've got some of these assays, we then need to test them to see if they'll even work. So we've got this data set. We need to actually empirically test the assays that we've created based on that data set. So here on the lower left-hand side, this is a qPCR amplification plot. And the only thing I really want you to take from this is we've got down here, this horizontal line on the bottom is the threshold line. And if one of these amplification curves here in orange crosses that threshold line, you essentially have a positive detection for the target of interest in that individual sample. And so what we did is we tested a number of these assays against blue mussel larvae, those various stages, as well as gametes, post-larvae, and the blue mussel adult. And then we also tested it against a number of off-target species, as we call them, like the oyster, soft-shell clams, as well as quahog clams. And so we were fortunate in that the Down East Institute happened to be rearing these other species at the time, and so we could test them against larvae and adults of other species. And so really what we're looking for here is assays that are only amplifying our blue mussel larvae, but aren't amplifying our blue mussel adults or any of these other species. And so once we have those, we can then move on to start doing some field sampling and field testing. So this is all great. We got these to work based on hatchery samples, but really the main goal here is to be using these with environmental samples, right? And so we teamed up with Bangs Island Mussels in Casco Bay and worked on a few of their leases. And I should say we are working on a few of their leases. This work is continuing to this day. And so these are the three that we've spent the most time at. Vast majority of the data I'll talk about today is from this Clappert Island lease that you see here in Casco Bay, this CAS CF3. And just want to give a shout out to Peter, Brittany, and Adam, who have helped me immensely with all this sampling. I absolutely could not do it alone. So a big thank you to them. But our sampling protocol goes as so. We first took a quantitative vertical plankton tow. So we essentially just lowered our plankton net down to the seafloor. Once it touches the seafloor, we then took a swath of the entire water column, which typically ends up being around 2,000, maybe a little bit over 2,000 liters in total that is filtered by that plankton net. Then we also did pump water sampling, and this is newer, that something that we really started doing in the past year. And this is based on some of Phil Yunn's work from the Down East Institute. He actually loaned us some of the equipment to do this. But we're essentially just lowering a weighted, or a hose with a weighted intake here down to 5 meters, pumping up 100 liters into a net on the boat, and then we can concentrate that tow that way. And once we have those samples, then we can collect our eRNA and eDNA samples. And we did so using a really simple kit that we've put together here based on a syringe. Here right to the right of the syringe is a Sterivex filter. This is just a little capsule filter you can screw onto the end of the syringe. So you can suck up your sample with the syringe, screw on that filter, push it through that filter, push your sample through that filter, then just add a little bit of preservative in this second smaller syringe, cap it, and that sample is done. And I should mention that we have to use a special preservative because of the volatility of RNA. So RNA degrades quite rapidly, so we need to preserve it on the boat. All right, so we took our samples, our plankton tow samples and our pump samples. We then took a subsample of that using those kits I just mentioned. The remainder of those plankton tows we preserved with ethanol, and this allowed us to then do microscopy counts for the preserved plankton tow, and then we could compare that back to our environmental RNA signal from those eRNA samples that we took. All right, and so what we're looking at here is a plot of the bivalve larval counts for our plankton tow. So remember, this is the vertical plankton tow. And these are taken at 3 different lease sites that you see up here. And so what you can see is down here we've got a little bit of activity. This started back in June 2023 when we first started taking our samples. And so in June we start to see it pick up through July, and then it ends up tapering off right around November, and then it those counts flatline. And so it really looks like they're pretty small numbers here. And then we start to see a pickup in June of 2024 again, and then we have a massive spike in August, which honestly we're still looking into. We then see a second peak here in October that drops down again in November. So we're seeing a kind of a similar pattern, save for this huge spike that we saw this past August. Now remember, I mentioned that we started doing the pump sampling within the past year. So we actually started this in June 2024. And so this is just kind of a side-by-side comparison of up on the top left-hand side, we've got the plankton tow samples from the vertical tow. And then on the bottom left, we've got the pump samples. The main thing I want you to take from this is, first off, if you look at the y-axes, there's quite a large difference there. But to simplify this a little bit, we see that in our pump samples, we've got, for instance, with this first sample, 7.4 larvae per liter versus 0.04 larvae per liter with our vertical tow samples. And so in other words, the bivalve larvae are much, much more concentrated at a depth of 5 meters than when you just take a swath of the water column. And so really we've shifted to this method of collection for right now because we're getting a lot more mussel larvae and a lot fewer of our off-target species. All right, and so here's just another plot showing those pump samples and the counts. This is just a bar plot in this instance. And if we overlay our eRNA signal, we can see that we've got a really high spike here in June that tapers off here in August and then picks up again where we saw that second spike in October. And then again starts to drop off. And so this is really cool because we're starting to see patterns with the 2 spikes. But what's interesting is you look at these bars in the middle here, and you're probably wondering what's going on with those. And what we think is going on is those are likely not mussel larvae. So there are bivalve larvae in the water column, but they're not mussels. And so we're actually in the process of doing this now, and over the summer we're going to be doing some of that metabarcoding that I talked about to get a better sense of what's going on with these samples here in the middle. All right, and so I also just wanted to touch on some of these other assays that we're working on, these LAMP-based assays that I mentioned. This stands for loop-mediated isothermal amplification. This is just a really simple way of saying you don't need a piece of specialized equipment like a thermal cycler to cycle up and down and up and down in order to amplify your target of interest. Instead, you can use this master mix here, this LAMP master mix, add your environmental RNA signal, or environmental RNA sample, and then you just have to incubate it at one temperature for 30 minutes. So much, much easier, right? And the cool thing is you can do this with something as simple as an electronic coffee mug that some of them actually will maintain 65 degrees Celsius. And so we've done this quite a bit, and it works really well. And then you get this really straightforward result where, again, if it's negative, you start off with pink, it's going to stay pink. But if you have your target of interest, it's going to turn this bright yellow color. And so you know, I've got larvae present in my sample. And so we've tested this with a bunch of those hatchery samples at this point that we collected earlier. And we're finding that we're only getting amplification for larvae and not any of the other blue mussel stages. And we're also not getting any amplification from those off-target species as well. All right, and so just a couple more slides here, and then I'll be done. So this is kind of the basic pipeline that we're imagining for these tools, where you collect your water sample using one of those kits, that syringe-based kit that I mentioned. And then you could use this LAMP-based assay to tell you whether or not you have mussel larvae at all in your sample. And if you want to get finer quantification, you could then, if you're a farmer, you could then either send it to us at Bigelow, and we could run it on our qPCR or dPCR systems. Or if you're a scientist and you're just interested in the larval ecology of your species of interest, you could certainly do that yourself. And this gives you an idea of not only the detection, but it'll allow you to get some idea of the quantification of the mussel larvae in your samples. And then finally, at least for farmers, we think that this could really have a big impact for informing spat collection. So if you could go out and test at your seed collection sites or on your lease site and determine when and where you actually have mussel larvae in the water column, then you have a better idea of the best times to collect, and that'll increase your chances of success for that particular season. And I should mention too, just really quickly, that this could be really useful for mussel spat biofouling mitigation too. Oyster farmers are constantly dealing with this issue of mussel spat just covering their oyster cages. And so knowing when that spat's around can help them to mitigate the work that and the cost centered around that. And then finally, this is my last slide. I'll run through this really quickly. And I just throw this up here because I actually just got back from a trip to New Zealand to go learn more about the green lip mussel industry that they have there. Very similar in the sense that vast majority of their spat comes from the wild. So they have to collect it from the wild. And so this is the green lip mussel that you see up here. And it actually comes up on this beach rack. So this is just a bunch of seaweed that washes up on this one beach on the North Island. And so it's really susceptible to issue, right? A couple of years ago, they had an extreme shortage here, and that wreaked havoc. And so there's more interest in trying to capture wild spat and to learn more about what the larval dynamics are around the New Zealand coast. All right, and with that, tons of people to thank up here, as well as a number of funding sources. And in the interest of time, I'll leave it there and take any questions if you have any.
Thank you, Dave. Questions? Says it's on.
Hello.
Hey,
Is it? Okay, it is.
I can hear you.
Hello. I was wondering what NGS sequencing was. It was on one of the slides, but I might have missed what it stood for.
Oh yeah, good question. I should have written that entirely out. Next generation sequencing. And so in this case, it's what we call Illumina sequencing, in which we basically take our RNA, break it up into a bunch of small pieces, and then sequence it.
Cool, thank you.
Yeah.
Other questions?
So what's the cost of your loop system in terms of individual cost to do one sample?
Just, you mean like for the collection of one sample or the entire cost?
Entire cost.
Yeah, it's a little hard to price out off the top of my head. Let's see. So the collection kits are about $25, $30. And then, yeah, I guess I can't say for certain because I'd have to factor in labor and the rising cost of a number of reagents. But in order of magnitude. How much, sorry? $100 or less? Yeah, I think it could be done for, yeah, $100 or less. Yeah.
That was super cool. Do you think mussel larvae are hanging out at 5 meters, like in its planktonic larval life history, or do you think they're migrating up and down the water column?
Yeah, that's a great question. So we chose 5 meters based on some of Phil Yunn's previous work down east. And he sampled at a number of different depths and ultimately settled for a lot of his work on 5 meters because that's kind of right smack in the middle of what the hypothesized vertical migration is. And so that's what we went with as well. We did take a few samples at 2 meters as well. And we saw similar abundances, but that could be due to coincidence, I guess. But yeah, this is purely based on previous work done here in the Gulf of Maine. Yeah.
Other questions? I have one for you, Dave.
Sure.
Is the goal to get to a point where maybe the farmers can do this themselves? Like I'm thinking with the LAMP assay in particular, like can an RNA extraction, is it then a cDNA synthesis from there or is it straight, right? Like I'm just wondering, can they get to the point where maybe they can do these sort of molecular things in the LAMP assay themselves?
That's the goal.
Okay.
Yes. And one of the big bottlenecks is, of course, the extraction process, right? And so we've been trying to figure out how we can do a crude RNA extraction that doesn't inhibit the LAMP reaction. And so there are a couple options out there. So far, the ones that we've tested, we've had a few issues with, only because the assay's based on pH, and it alters the pH, and so you don't get that nice color change. However, I should mention that there's been a lot of really cool progress in the LAMP field recently, where they've developed some really low-cost, like smartphone enabled devices. So they're DIY, and you can do a fluorescence-based assay that I think would work really well with that. And so that's part of our plan moving into the future is building a couple of those and seeing if we can't do that with fluorescence-based assay.
Any other questions? All right, let's thank Dave again. Thank you.
Yeah, thank you.